Welcome to Catch This. My name is Trudy Ray, and today I would like to address the conspiracy theories that claim that SARS-CoV-2 is man-made in a lab, or that even if it wasn't, that it probably existed in a lab and escaped from there. In this video, I will provide scientific evidence that shows that it is virtually impossible for this virus to have been engineered in a lab, or even have escaped from a lab, and that it most likely arose from natural recombination of different coronaviruses in a non-human animal. Very early during the pandemic, Christian Anderson, a scientist specializing in genomics, evolution, immunology, and infectious diseases at the Scripps Research Institute, published this paper in collaboration with others, in which he highlighted two notable genomic features of SARS-CoV-2. The first feature is that SARS-CoV-2 appears to be optimized for binding to the human receptor ACE2. And the second feature relates to the furin cleavage site in the SARS-CoV-2 spike protein. We'll come back to the furin cleavage site, but let's start with the optimized binding feature. SARS-CoV-2 has this highly immunogenic protein on its surface called the spike protein. I have discussed it in many previous videos. A portion of the spike protein, known as the receptor binding domain, binds to a receptor protein on our cells called ACE2, and this binding allows the virus to enter our cells. We know that there are 6 amino acids in the receptor binding domain that are essential for binding to ACE2. Here they are: 1, 2, 3, 4, 5, and 6. So let's look at this logically, perhaps from the point of view of someone with the intention of engineering a new virus that infects humans and transmits from one human to another and another and another in a sustained manner. Logically, you would start with the most closely related coronavirus. The most closely related coronavirus by sequence similarity that we know of is called RATG13, and it only infects bats. A scientist with this evil intent would most likely have selected this virus and would have introduced mutations to allow it to bind to our ACE2 receptor so that it can infect humans. However, there's a slight problem here. 5 of the 6 essential amino acids in SARS-CoV-2 differ from the amino acids in the receptor binding domain of RATG13. The only way a conspirator scientist would have known exactly which mutations to introduce into the receptor binding domain of RATG13 to enable it to bind our ACE2 receptor would have been by predictive computational analysis, since crystal balls are not very scientific. But as it turns out, computational analysis does not predict that the current 6 amino acids in the receptor binding domain of SARS-CoV-2 would enable high affinity binding to our ACE2 receptor, as shown by this publication and others. In fact, the SARS-CoV-2 receptor binding domain sequence is different from that shown to be optimal for receptor binding in SARS-CoV, the original SARS virus from 2002. Computer programs predict that the SARS-CoV-2 receptor binding domain actually shouldn't really bind very well to ACE2 receptor at all. However, in reality, the binding between ACE2 and these 6 amino acids in SARS-CoV-2 is tight. This is already pretty strong evidence that SARS-CoV-2 is not the product of purposeful manipulation, and that this high affinity binding of the SARS-CoV-2 spike protein to human ACE2 is most likely the result of natural selection. But there's more evidence that this virus could not have been made in the lab. When you do an alignment of the RATG13 nucleotide sequence against the SARS-CoV-2 nucleotide sequence, you can see that there is about a 4% difference between these two sequences. So if you do the math and you take into account that these two genomes are almost 30,000 nucleotides in length, then that comes out to about 1,200 nucleotides that are different between these two sequences. In this alignment, with the query sequence representing RATG13 and the subject sequence representing SARS-CoV-2, everywhere you see a gap where you don't see a line joining the matching nucleotides, there is a mismatch. So there are about 1,200 mismatches, and as you can see, they're scattered all over the genome. They are not just limited to the receptor binding domain or to other domains that might be critical for enhanced infectivity and transmission. Why would someone make all those changes across the genome? If you're a virologist looking at this, it just doesn't make any sense. That is a lot of work, especially considering that one wouldn't really know what effect each of these changes would have on the function of the virus. Another argument made by lab leak conspiracy theorists points to the furin cleavage site in the SARS-CoV-2 genome. This is a sequence in the SARS-CoV-2 genome that is not present in RATG13. It's not present in the first SARS coronavirus. It's not present in any of the coronavirus sequences listed here. Although there are other human coronaviruses that do have a furin cleavage site, like MERS coronavirus, OC43, and HKU1. The inserted nucleotides code for this PRRA sequence, but ultimately this change results in these amino acids RRAR next to each other, which are recognized by our cellular protease called furin, which cleaves the site. This cleavage by furin is required for infection of cells. I know that at first glance you look at that insertion and think, whoa, that looks suspicious. But I want to caution that insertions like that happen all the time in nature, naturally. This is not uncommon. Viruses mutate and they recombine all the time. New sequences are inserted into viral genomes all the time, randomly. Coronaviruses are highly combinogenic. This means that when two different coronaviruses infect a cell, their genomes can recombine and out come progeny with pieces of genomes of either parent. I guess you could kind of think of it as viral sex. But let's take a closer look at the sequence, particularly at the underlying nucleotide sequence that codes for these amino acids. When you do, there's something odd that would stick out to any molecular biologist or anybody who has spent any significant amount of time introducing mutations into DNA. As I have discussed on previous episodes, genomic DNA ultimately codes for protein in nucleotide triplets, with each triplet of nucleotides coding for one amino acid. That's called a reading frame. Usually, when one wants to insert a mutation in DNA that ultimately results in insertion of several new amino acids, but keeps the rest of the protein sequence the same, one would introduce this mutation at the DNA level in frame, meaning that you insert your new sequence after a complete previous triplet. But here, as you can see, the 12 nucleotide insertion shown in red that leads to the PRRA amino acid sequence is out of frame. The 12 nucleotides in red are inserted between this C right here and this A right here, relative to the RATG13 genome. Here's the C and here's the A in the SARS-CoV-2 genome, relative to the RATG13 genome. Such an insertion breaks up the reading frame and could potentially change all the amino acids following the sequence, potentially changing the protein and changing its function. You can ask any experienced molecular biologist. Nobody would insert a mutation like that. In fact, it kind of looks like a rookie mistake. But wait, there's more. This resulting cleavage site, RRAR, although it is sufficient for the success of SARS-CoV-2 as a pandemic virus, is considered a suboptimal furin cleavage site. The presence of multiple basic amino acids like arginines is what defines a furin cleavage site. However, based on established literature, a furin cleavage site that is considered optimal typically consists of the sequence arginine, any other amino acid, a lysine or an arginine, and another arginine. So tell me, why would someone with the intention of engineering a virus that infects humans optimally and results in optimal human-to-human transmission insert a suboptimal cleavage site? But wait, there's even more. The addition of the proline to that sequence is predicted to result in the addition of O-linked glycans to this serine, this threonine, and this serine. O-linked glycans are sugar molecules that are preferentially added to serines or threonines under certain conditions, and this addition, known as glycosylation, typically serves as a protective shield from the host's immune system. Importantly, this suggests that the acquisition of this feature resulted from interaction of the virus with an immune system, once again ruling out purposeful manipulation of the viral sequence. Long story short, no experienced virologist would have inserted that 12 nucleotide sequence there in that way to result in the present suboptimal furin cleavage site, or even have known that this would result in a functional virus. We don't know what makes this virus transmissible in humans. We don't know what controls its pathogenesis. So if you wanted to create something that has these characteristics of efficient human-to-human transmission and this level of pathogenicity, you wouldn't know how to do that. There isn't anybody on Earth who knows that. And then on second thought, if you were to design another SARS virus, wouldn't you start with the first SARS coronavirus? The one from 2002? Just a thought. I also want to add that the overall structure of this virus is distinct from the coronaviruses we know of, and instead most closely resembles viruses found in bats and pangolins that have been little studied and have never been known to cause humans any harm. Okay, so some people say that even if it wasn't engineered using genetic engineering methods, the virus could still have been generated by repeated passage in cell culture or animals in a lab. It is true that passage through cell culture or animals can lead to mutations. However, passage in animals would give you an idea of the fitness of the virus in that animal, but you still wouldn't know how well it works in humans. Also, SARS-CoV-2 has been mutating at a rate of about 20 nucleotides a year during this pandemic. So if one were to start with RATG13, the coronavirus with the closest known sequence identity to SARS-CoV-2, how many years would it take to get a difference of 4% between two viruses of this size? Let's see, to reiterate. A 4% difference between two viruses, 30,000 nucleotides in length, means that about 1,200 nucleotides are different between these two viruses. 1,200 divided by 20, that's 60 years. For a project this complicated, you would probably need a pretty large group of people to help out at various steps of not only the initial experiments, but also the inevitable cover-up. So you're trying to tell me that a large group of scientists kept this project going and secret for 60 years. Didn't some famous guy once say that 3 can keep a secret if 2 of them are dead? I can't remember who it was, but I think he had a point. Okay, so let's say that it wasn't engineered, but it did exist in the lab in Wuhan and it escaped somehow. There's a problem with that argument too. There are 2 virus lineages, lineage A and lineage B, that split very early in the pandemic in the city of Wuhan. At that time, there was only a 2 nucleotide difference between lineage A and lineage B viruses. All the cases identified at the Huanan market in Wuhan were infected with B lineage virus. There are other cases that resulted from infection with A lineage virus that were linked to other markets. That means that any theory on the origins of the virus has to account for not just the linkage to the market, but the linkage to the different lineage at the different markets. If the virus emerged through the wildlife trade, that's pretty easy to explain. There used to be these huge wildlife farms in China that were shut down in February of 2020. These wildlife farms were a $70 billion industry that employed about 14 million people. These farms bred exotic animals like civets, porcupines, pangolins, raccoon dogs, and bamboo rats in captivity, and then supplied these animals to vendors at the various markets in Wuhan, which is a distribution hub for a lot of different types of commerce. So if there was an animal or a group of animals at one of these wildlife farms that was infected with the original SARS-CoV-2, then maybe one animal passed it to another, and that 2 nucleotide change from lineage A to lineage B could have easily occurred when the virus jumped from one species of animal to another. Then these animals were brought to the markets, and an animal or 2 that was infected with lineage A virus ended up at one market, and another animal or 2 infected with lineage B virus ended up at the Huanan market. This is likely. What is not likely is a lab leak scenario where 2 different workers became infected with different lineages and then went to different markets and were successful in spreading the virus there, considering that most initial jumps of a new virus into a human end up with that human and are not successfully transmitted to other humans. Or a worker became infected, went to a wildlife market, infected some people there, and then went to another market. And in the meantime, the virus acquired these 2 mutations to become lineage B, and then that person infected more people at the second market. That is very unlikely. There are just too many convenient coincidences needed for a lab leak scenario. Look, there are documented instances of laboratory escapes of viruses, but in the case of SARS-CoV-2, there are too many separate lines of evidence that suggest that SARS-CoV-2 acquired these mutations via recombination or mutation. Keep in mind that the emergence of new viruses and the emergence of conspiracy theories regarding their origin seem to go hand in hand. This is no new thing. Past pandemics have likewise been accompanied by conspiracy theories and waves of mistrust in science. It happened during the 1918 flu pandemic when, much like now, dangerous misconceptions and conspiracy theories regarding the origin of the virus soon emerged. One myth linked the pandemic to the use of aspirin produced by the German pharmaceutical company Bayer. Another claimed it was the result of biological warfare. Many people feel very uncomfortable dealing with the unknown, and as something new happens, they want answers. In fact, there's a certain comfort in conspiracy theories because it would mean that somebody's actually in charge. Many people feel that even if the people in charge are malevolent, that's better than acknowledging that we live in a chaotic universe that just doesn't give a damn about us, which is terrifying, but is actually true. Nature wants to kill you. In contrast, scientists are used to the unknown. We are completely comfortable with not knowing. In fact, that is what drives us to find evidence-based answers. One more thing. Who wants to bet that when this pandemic is over, there will be new conspiracy theories, that this pandemic never even happened? Thanks again for listening. My name is Trudy Ray. I'll see you next time on Catch This.